Hey students, today we're going to be doing the Plasmolysis lab. You'll need a small piece of Anacris or Elodea, which is an aquatic plant. So carefully remove one leaf from the plant and place it onto your slide. You may want to add a drop of the water from the container to help hold the leaf in place, and then carefully take your cover slip at a 45-degree angle and lower it down until you release all the air from underneath the cover slip and you have a wet mount. Next, you are going to place your slide on your microscope. Focus under scanning power, get it into focus using your coarse adjustment, then go to low power and you should be able to see all these tiny little cells. Each one of these represents one cell. Inside of each cell are the green chloroplasts. My coloration on the monitor isn't the best, but once I get to high power, you can see with a little bit more detail. And so here we're looking at the cell walls, and we have lots and lots of chloroplasts that are filled within the cells inside. So now what we're going to do is we are going to expose these cells to a hypertonic salt solution. So we are going to place the cells in a salt solution, and I will show you how that works next. While viewing your cells underneath high power, you should place several drops of 10% salt water next to the cover slip. So the water is going to then be pulled underneath the cover slip by the addition of a piece of paper towel. You can see the water wicking through here, pulling out the normal water and pulling the salt water underneath the cells, underneath the cover slip, exposing the cells to a hypertonic environment. So as you watch, you might be able to see the cells start to plasmolyze. I am currently adding that salt water to the cover slip. I lose focus a little bit. I'm going to try to keep it in focus so you guys can see this. And so as the water starts to get pulled through or underneath the cover slip, we should be able to observe some plasmolysis happening. So notice the chloroplasts are kind of arranged around the outside of the cell. Eventually, as this water leaves the plant cells, we're going to start to see this shrinking occurring. So there you have it. You can kind of see it if I keep this in focus, how the cells are starting to contract. All of the water is being evacuated because of osmosis, and the cells are going through plasmolysis. So the more water I can pull out and the more salt water that enters in, the better we're going to be able to see these plasmolyzed cells. So again, this is under high power 400x, and you can start to see the cells going through plasmolysis. And let me just pan down a little bit, and you can see some of these over here, like right here in the center. These cells are good examples of cells that have gone through plasmolysis. So this line right here, you can see the cell membrane. The cell wall does not shrink. You can see that cell just kind of filling back up with some water, and now all of a sudden you're going to start to see some more water evacuating. I'm going to add a little bit more salt water to the edge of the cover slip, and we're going to see if we can get even some more plasmolysis to happen. But if you do this correctly, you're going to end up seeing all the cells where the cell membranes have contracted and the chloroplasts become centrally located. So basically, all that water that's in a vacuole and inside the cell cytoplasm will exit the cell. It's going to move from a high concentration to a low concentration, and this is plasmolysis. So in the real world, you might see a plant exposed to an environment where there's not much water on the outside. The plant will tend to wilt. They'll get very soft, and they'll start to lose water. And this right here, folks, is plasmolysis. Pretty cool, isn't it? I thought so. So the last part of the experiment is to place the cells back into a hypotonic environment. So I'm currently adding some more water to the edge of the cells, and I'm adding 100% H2O. So just all distilled water, dropping it next to the cover slip. And as I'm doing that, there's a lot of movement happening, so it's hard to see the cells. I got to keep fine focusing. But as I pull that salt water out with the paper towel, what we're going to see is the water flowing back in. Sorry there, I lost it. The water's going to flow back in, and we should be able to see the cells fill back up with water. So those are all plasmolyzed yet. Let's see if I can get some more water pulled out here and get that distilled water. There we go. So watch, they're filling back up with water. Some membranes are starting to fill up. They're starting to expand. So the cells are becoming much more turgid, full of internal water pressure, simply because